All right, so we left off talking about, or wrapping up the different processes that are going to occur in cellular respiration. We talked about glycolysis, which is used to take sugar molecules and it's to break them in half in order to make pyruvate. And then that pyruvate gets converted into acetyl-CoA. And then that acetyl-CoA will enter into the Krebs cycle where it will remove all these carbon dioxide molecules. And then finally, you're going to go through the oxidative phosphorylation, which is where you're going to generate all your ATP. And so this is a perfect summary slide as far as information that you need to know for cellular respiration. All right, remember I said you need to know the starting products and the end products. All right, so for example, in the Krebs cycle, you need to know that 2 acetyl-CoA molecules are going to enter it, and that you're going to lose 6 NADH, 2 FADH2, 2 ATP, and the only thing that's not on there is carbon dioxide. There should be 4 molecules of carbon dioxide is also a product of the Krebs cycle. And so just to go through these again, because I'm sure y'all could probably use it. So remember the equation for cellular respiration is glucose plus oxygen. And that's going to yield water, carbon dioxide, and energy. And so remember, we're taking electrons away from glucose and we're giving them to oxygen. Once you add on those electrons, that oxygen is going to become water. Once you remove those electrons from glucose, that's going to become carbon dioxide. And so we're just shifting electrons around. And so the first process, remember we have glycolysis, and that's going to start with glucose, and it's going to be converted into 2 pyruvates. Those 2 pyruvates will go through this transition stage where the 2 pyruvates are going to be converted into 2 acetyl-CoAs. And then those acetyl-CoAs will enter into the Krebs cycle. So that's going to start with the 2 acetyl-CoA. And at the end of the Krebs cycle, you're going to end up with carbon dioxide, ATP, NADH, and FADH2. And then after the Krebs cycle, you go into oxidative phosphorylation. The first one, the electron transport chain, that's where you're starting with all of these electron carriers, NADH and FADH2, and they're going to drop off their electrons to oxygen in order to make water. And we're also going to make some ATP in the process because of that chemiosmosis that's occurred. And then finally, after the electron transport chain, we have this chemiosmosis, and that's using the proton gradient that was a result of the electron transport chain. It's using that proton gradient in order to power ATP synthase, which is lodged in the membrane. And so chemiosmosis, you're going to end up with 32 to 34 ATP. So that's just kind of a brief rundown of what we talked about in class. And again, I think this is a really good start studying here when you're studying cellular respiration. This tells you where all the processes are located, how much ATP they make, what they start with, how many NADs and FADs they make. So start with this, but don't forget to add the carbon dioxide to the Krebs cycle, which I should have added to the slide, but I just now noticed it's not there. So in addition to the Krebs cycle, the products are going to be 2 ATP, 2 FADH, 6 NADH, and 4 carbon dioxide. All right.
It's products of Krebs.
Yes, that's Krebs. All right, do we need to go over this again, or are we ready to move on? Somebody tell me something. Next? Okay. All right. Get it over with? Okay. All right, so we've kind of wrapped up cellular respiration. And like we said last week, we're going to focus on the aerobic aspect. And remember, aerobic respiration and cellular respiration are the exact same thing. But we also have respiration that can occur without oxygen. And so we call that anaerobic respiration. Anaerobic means without oxygen. And so various different organisms are capable of utilizing anaerobic respiration. One type of anaerobic respiration is going to be fermentation, which we're going to discuss. And we said there's two types of fermentation. You've got lactic acid fermentation, and then you've got alcohol fermentation. And so if you don't have any sugar molecules that can be used to generate energy, there's chemical pathways that exist in our body that can break down other macromolecules in order to provide energy. So the one we've been focusing on is right here in the middle, cellular respiration. But that's if only you have a lot of sugar in your diet or glucose. Let's just say that you're anorexic and so you're not eating anything. Then if you're not eating anything, you're not going to have any of those sugars to digest. So what's going to happen, your body has to generate energy or else it's going to die. And so the way that it's going to generate that energy, it's going to take your muscles. It's going to take the protein that you have built up in your muscle, and it's going to break that down to use energy. And so that's what this pathway over here is showing. So even though you might not necessarily have sugar, there are similar pathways that are used, which are still going to be used to make energy. And so that's why people who are anorexic, their muscles atrophy because your body's breaking them down because it has to generate energy from somewhere. So it can take it from the proteins. It can also take it from fats. Fats store a lot of energy. Yeah.
Atrophy?
Atrophy means like kind of waste away. So people who are like in a hospital bed for 6 months or something, their muscles will atrophy because they're not using them. And so you can use either proteins or you can use fats. And so both components of the fats, both the glycerol and the fatty acids, will feed into various different cycles in order to still produce energy. So I don't necessarily want you to memorize this or anything. I just want you to know that there are other ways to break down molecules in order to generate ATP. You can use proteins or you can use fats. But carbs or the sugars, it's going to be the most efficient way. That's going to produce the most energy. But if that's not available, then you can use one of these alternative pathways. All right, so now we're going to focus on anaerobic respiration. And like we said, both aerobic and anaerobic, they're all going to use glycolysis. Glycolysis is a universal thing. Every single living organism uses glycolysis. And so from glycolysis, again, we get 2-pyruvate. We generate a couple of electron carriers right here. And so if it's going into normal aerobic respiration, it'll enter in the Krebs cycle. It'll produce more electron carriers and some other things. It'll go through the electron transport chain. And remember, that final electron acceptor is oxygen. So when we finally give those electrons from glucose to oxygen, we're going to make water as a byproduct of aerobic respiration. But in anaerobic respiration, you're still going to go through the Krebs cycle. You're still going to go through the electron transport chain. The only thing that's different is that final electron acceptor at the end of the electron transport chain is not going to be oxygen. Sometimes it can be nitrogen, it can be a sulfur, it's just something else other than oxygen. So when you're talking about anaerobic versus aerobic respiration, all you're talking about is the electron acceptor at the end of the electron transport chain. So it's going to be oxygen in aerobic and something other than oxygen in anaerobic. Now, let's just say that organisms might not be able to use either one of these pathways. Well, there's still an alternative pathway that's called fermentation. And so fermentation, again, is going to use glycolysis, but it's going to bypass the Krebs cycles and the electron transport chain and oxidative phosphorylation. Instead, what's going to happen is it's going to take those 2-pyruvate molecules and it's going to convert it to either lactic acid or into alcohol. And so that's where we get the lactic acid fermentation and alcohol fermentation. And our bodies can actually perform fermentation. So if you've ever been at the gym or you go out for a run or something and you run 6 miles and your calves start to burn, you feel that burning sensation, that's actually lactic acid building up in your muscle tissue. And so what happens when we're working out, we're still breathing, we're still taking in oxygen, but we're not bringing in enough to be able to utilize cellular respiration efficiently. So our bodies actually switch, or not necessarily the bodies, but the cells that are being utilized the most, that are using the most oxygen, they'll switch to fermentation. Because they're still going to have glycolysis, and they're still going to have this pyruvate to work with. But instead of going down the aerobic respiration pathway, because we're breathing really heavily and we can't breathe in enough oxygen to keep up with the demand, it'll switch to fermentation. And so that pyruvate will be converted into lactic acid. And it's that lactic acid that gives you the burn whenever you work out or use your, exhaust your muscles. And so other organisms capable of using fermentation are going to be yeast, and bacteria as well also use fermentation. All right, so we're going to go and we're going to talk about one of these individually. And so organisms that ferment, like bacteria for instance, they always use fermentation. All right, they don't use cellular respiration or aerobic respiration. They're always going to use fermentation. And so fermentation is, again, going to produce either alcohol as a byproduct or lactic acid as a byproduct. And again, you're taking this pyruvate molecule that you get from glycolysis, we're still removing electrons from it, but we're giving those electrons to help regenerate our NAD positives. Remember, our NAD positives are our electron carriers, and they're going to be used in several different processes. So it's important that we kind of re-up them and keep them regenerated for all the necessary processes. And so both of these, lactic acid and alcohol fermentation, are going to be kind of similar to one another. So if you look at alcoholic fermentation, and here's an example, making beer or whiskey or whatever type of liquor you want to discuss, or even wine. But anyways, you're still going to go through glycolysis, so you're still going to make your 2 pyruvates. But instead of entering into the Krebs cycle and oxidative phosphorylation and all that, it's going to bypass all that. And from that pyruvate, you're going to release some more carbon dioxide, so similar to what the Krebs cycle, because in the Krebs cycle, you're starting with pyruvate, and you end up removing some carbon dioxide, so it's similar to this. In addition to creating these carbon dioxides, you're also going to regenerate these electron carriers. So starting at the beginning of fermentation, you're going to start with the NAD positive. And then after both types of fermentation, you're going to end up with a couple of molecules of NADH. And then you'll also generate 2 ATP. So for alcoholic fermentation, your products are going to be 2 NADH, 2 carbon dioxides, and 2 ATPs. And so, whereas if you look at lactic acid fermentation, it's still going to use glycolysis, it's still going to start with pyruvate, it's going to bypass the Krebs cycle, and the electron transport chain, and all that. And it'll immediately be used to generate 2 molecules of ATP, 2 molecules of lactic acid, and then 2 molecules of NADH. So they're very similar processes, and they're going to occur in different types of organisms. In alcohol fermentation, you're going to end up with alcohol, carbon dioxide, NADH, and a little bit of energy. And then for the lactic acid, you're going to end up with 2 lactic acid molecules, 2 ATP, and 2 NADH. So when you compare the efficiency of fermentation to cellular respiration is way more efficient, which is why our bodies don't use fermentation all the time. So this fermentation is just kind of a backup mechanism in case we need it. Our bodies and our cells, they have tons of backup mechanisms. There's always a plan A, B, C, D, and E. And so this is plan B for if we're exerting ourselves and can't get enough oxygen into our cells. Questions?
Yes, could you explain what the process out the alcohol and the
Yeah, so you're going to start with 2 pyruvates. And you're going to start with 2 NADH. And so after this, because remember in the Krebs cycle, this is eventually going to be converted into that. So you're still stripping electrons away from this and generating carbon dioxide. And so the output, you're going to generate 2 ethanols, which is a type of alcohol. You're going to generate 2 carbon dioxides. And then you're going to regenerate these 2 NAD positives. Whereas in the lactic acid fermentation, you're starting still with 2 pyruvate and 2 NADHs. And then that's being broken down and rearranged to make 2 lactic acids, 2 NAD positives, and 2 ATP. For some reason, they didn't include ATP in the output, but both of them make 2 molecules of ATP. Any other questions? Yes?
Why does the alcoholic,
That's good.
why does the alcoholic fermentation release carbon dioxide while the lactic one doesn't?
I don't have a good answer just because
Okay.
that's the way it is. But that is why you can't use, I mean, that is one of the reasons why we use alcoholic fermentation to make, alcohol, is because of that carbon dioxide. So these bacteria or these yeast or any other organism that's using the alcoholic fermentation, they're always going to be releasing carbon dioxide. So if you've ever made your own beer or wine, or, there's lots of bubbling, right? There's lots of carbon dioxide, which is why when you have a beer, there's all these bubbles in it, right? It's carbon dioxide that's being released. And so, but as far as why that one and not this one, I don't know. It's just a different process. Yeah, you had a question.
Is the pyruvate, sorry if this is already been discussed,
That's
but
fine.
is the pyruvate is just dividing the glucose molecule?
The glucose, so you start off with glucose, and then after glycolysis, you're going to end up with 2 pyruvate. So glucose is a 6-carbon sugar, and each one of these is a 3-carbon sugar. So you're basically just breaking glucose in half and generating 2 pyruvate molecules. And then the pyruvate, again, like we said, this process, this will be used in every single cell, in fermentation, and aerobic respiration, anaerobic respiration. So everybody's making pyruvate from glucose.
And that's glycolysis.
Yeah, glycolysis is the process. It starts breaking down sugar. Remember we said that glucose is going to be oxidized. Anytime you oxidize something, you're taking electrons away from it. So we're oxidizing glucose, and once you take all those electrons, you're going to end up with carbon dioxide. And so glycolysis starts this oxidation process. So you finish breaking down glucose in the Krebs cycle. So glycolysis and the Krebs together, their main function is to oxidize glucose. So half of it gets done in glycolysis, and then it finishes it up in the Krebs cycle. And that's why once you totally oxidize it, you're ending up with carbon dioxide. So you know that you've finished breaking down glucose once you release all that carbon dioxide in the Krebs cycle. So the Krebs cycle completes the breakdown of glucose. And then after the Krebs cycle, and we've loaded up all of our NADs and our FADs, that's when we go immediately to the electron transport chain. So the whole point of glycolysis in the Krebs cycle is to break down glucose and remove those electrons from them. So that's what's happening in both of these. Then once you have all those electrons, then you move into the electron transport chain where you hand off those electrons to to oxygen in order to make water. Other questions?
Okay, so once it's totally oxidized, the glucose then will become CO2. What was that second part that you said right after that? want to make sure. After Krebs, it starts the electron transport.
Yeah. So remember the whole point, all we're doing, we're taking electrons away from glucose, and when we're taking them away from glucose, we're using glycolysis in the Krebs cycle. So that's the first part, removing the electrons. And the second part is giving them to oxygen. And we give them to oxygen through the electron transport chain. All the NADs and all the FADs that we generated in glycolysis in the Krebs cycle are going to go to the electron transport chain, drop off those electrons. Those electrons will travel down and meet up with oxygen to make water.
And then that third step.
And then chemiosmosis, remember chemiosmosis is related to the electron transport chain, because as you go down the electron transport chain and those electrons travel down to meet up with oxygen, remember you're pumping a lot of these hydrogen ions outside into the inner membrane space. So this is the space between the outer and the inner membranes. And so as a result of the electron transport change, you get all these hydrogen ions that are outside. But remember, you already had a whole bunch of hydrogens out here. So there's already a high concentration outside and a low concentration inside. So you use a little bit of energy to force them against the gradient. But now that they're out there, now they're just going to want to come back in, right? So they're going to turn around and they're going to come back in. But when they come back in, they're going to go through this ATP synthase. And so it's this ATP synthase which is going to generate, it's going to take a phosphate group and add it to ADP in order to make ATP. So you might be wondering, when it leaves, why doesn't it just go through this in the first place? Well, the reason why is